Welcome to our fifth and final webinar in a series that focuses on improving immunohistochemistry and immunofluorescence workflows. Today's webinar is about selecting IHC detection reagents. This will be presented by my colleague, Dr. Craig Powell. Following the 20-minute presentation, we will have 10 minutes for a Q&A session. Please submit any questions you may have using the chat function on the GoToWebinar site, and we will answer your questions accordingly. I am now pleased to introduce you my colleague, Dr. Craig Powell. Over to you, Craig.
Thanks, Byron. Hello, everyone, and good morning, good afternoon, and good evening, wherever you may be located around the globe. Thank you for joining us for this fifth and final webinar in our current series. Our focus today, as Byron mentioned, is on choosing immunohistochemistry detection reagents. Okay, here's a brief overview of what we'll be discussing in the webinar today. We'll look at a couple of commonly encountered situations that we field in technical service at Vector Labs. The first one, those investigators who may be getting into immunohistochemistry or those who may be looking to simplify their current procedural workflow. Then we'll look with investigators that may have an existing methodology, but maybe they want to expand it or augment it, or there's some change that they want to slot in there, and we'll look at some of those commonly encountered scenarios. Then we'll have a quick discussion on enzyme substrates and close out the webinar with some cost considerations. Okay, so this is one scenario that we do encounter in technical service quite often for those investigators who might just be getting into immunohistochemistry or trying to introduce it into the lab. They want to identify a target antigen in a given tissue specimen. More than likely, they have an idea, of course, on what that target antigen is, and they probably have the specimen already in hand, probably already processed or blocked. And they're asking us for insight into how do I select detection reagents. More than likely, at this stage, the investigator has already done a bit of research and probably have a primary antibody in mind, whether it be a mouse or a rabbit, for example, primary antibody to detect that target antigen. This is also, this given scenarios, investigators who might be struggling with reproducibility or consistency with their existing methodology. Maybe they're a little frustrated in sourcing reagents from multiple vendors, trying to match reagents, and it's been a very time-consuming optimization process. So they do reach out to us and ask for inquiries and help on how do I simplify my immunohistochemistry. Now, a couple of questions that we certainly ask at Tech Service number one, what's the species of the primary antibody? What was it raised in? And this helps sort of set the scene on establishing what detection or secondary detection reagents are required. For example, you go have a mouse primary antibody, you require an anti-mouse detection methodology. The second question that's important to establish is what's the species of the tissue specimen? And certainly this helps identify any potential cross-reactivity or species cross-reactivity that may arise when placing on an antibody, a secondary antibody, onto a given tissue specimen. And importantly, the third question we do ask, and certainly investigators should be asking themselves, is whether the primary antibody recognizes the target antigen in that given tissue specimen. And I've got a couple of points down the bottom here. If they say yes, well, there's a good chance of success. And I've got in parentheses there immunohistochemistry, and that's really a prompt for me just to certainly say, has it actually been determined to work in that tissue specimen using the application or the technique of immunohistochemistry? And at that point, some investigators hesitate and say, you know what, it's actually been used in Western blotting. That to me would be a bit of a question mark. It hasn't actually been shown to work in the application of immunohistochemistry. They are different applications, obviously, and just because it works in Western blotting doesn't necessarily mean it's going to work for immunohistochemistry. So you may want to visit whether or not that would be regarded as an unknown, in which case you'd want to ensure the availability of positive control material you can run in parallel to make sure that you are getting adequate or appropriate staining. And it's certainly, if the answer is a dead stop no, you're going to have to reevaluate the primary antibody selection, go back to the drawing board, pick another primary antibody that is known to work in that tissue specimen. Okay, so once we've got that, we certainly do recommend a very straightforward methodology, such as the cartoon images that I have here, whereby you have an unconjugated primary antibody that are bind to a target antigen, and we recommend using something like one of these ready-to-use enzyme polymers, which would be like, for example, anti-mouse secondary antibody polymer with the enzyme conjugated to it, bind into this mouse primary antibody, and then you detect with an enzyme substrate, such as, for example, DAB. This simple methodology, it hits all the checkpoints as far as I'm concerned in terms of simplifying immunohistochemistry, or for those who want an introduction into immunohistochemistry in their lab. It hits the points for sensitivity for moderate to highly expressed antigens. It's an uncomplicated, one-step and relatively fast or quick detection methodology, whereby after the primary antibody has been applied, you can simply apply that secondary detection reagent, in most cases for 30 to 60 minutes, develop the substrate, and you're done about an hour after applying that primary antibody. And certainly with some of these reagents, you can get them as ready-to-use reagents and also incorporate as part of a complete or a comprehensive kit format. And certainly those 2 points that I'm highlighting there, being ready to use and comprehensive kit formats, certainly help substantially to reduce optimization requirements for the end user, saving time and cost, particularly labor costs or hands-on costs, trying to optimize concentrations and dilutions and things like that. So certainly this would be a great segue for those looking to simplify immunohistochemistry, or indeed trying to looking to introduce it into the lab. Indeed, a lot of investigators may be part of a larger study whereby immunohistochemistry is only one small segment. They're just looking for the expression of a given gene, or a protein, I should say, after a gene's been introduced. So this would be a great way to look at that. And these kits do exist out there. I'm just making you aware of the fact that immunohistochemistry in a box is a saying I've used here, whereby you just simply add primary antibody. So this is an example of, well, these are examples of some of the complete or comprehensive kits, whereby it includes an enzyme quenching reagent, a general protein block, a secondary detection reagent with the enzyme conjugated to it, and a substrate. So this is a complete kit format. Now here's a couple of pros and cons with these comprehensive kits. Number one, a great pro or advantage is their volume matched, lot specific kit formats. You're not scrambling to try and grab reagents from different vendors and sort of piece it together. So it reduces optimization in that regard. But what it does do, unfortunately though, it does reduce the flexibility of the detection methodology if you're going to change out some component, or at least look to change out a component. But nonetheless, these are a great segue into immunohistochemistry. A quick word about some of the enzyme polymer reagents out there. They're not all created equal, and they do vary, in some cases considerably, with their sensitivity and level of background that they generate. And what I've got here is a couple of serial sections, one here and one here, staining for the antigen P53. And you can see in this image, with I've got the red dot highlighting, some very robust nuclear staining for P53, which is great, but it's also accompanied by a substantial amount of this cytosolic, I would call, background or toning staining, which is certainly contributing to some of the intensity of the staining in the nuclei. Over here, the corresponding polymer system, you do still see some nice nuclear staining for DAB here, but it does not have that cytosolic background staining. So certainly just keep that in mind. It does depend a little bit on the procedure you're looking for, the scope of the project. So if sensitivity is a concern or you need clean results, certainly just be aware of the fact that swapping out some of these detection reagents, or at least modifying it a little bit, can give you cleaner results. Okay, so we've just looked at how you may want to introduce immunohistochemistry into your methodology or into your lab, or simplify it. How about the investigators out there that have an existing system? And certainly I will say, for those listeners out there who do have a methodology in place that is giving adequate sensitivity, it's reliable, giving consistent staining, I'm going to say, you know what, I don't have a compelling reason to tell you to change. That's great. Keep it going and keep churning out those great results. But I will say that a lot of research projects are not static, and certainly biology is not static by any means. And at some point, yeah, there may be a requirement to change the existing detection methodology. Perhaps there would be a requirement to increase sensitivity, look at a different tissue type or a different species of the specimen, maybe introduce a different primary antibody, or maybe explore introducing a second or third primary antibody through multiple antigen labeling like multiplexing. Indeed, the last bullet point I've got there is some investigators who are a little frustrated with the poor signal-to-noise ratio with their existing methodology. And I'll just refer back to the previous slide I showed you with those differences in sensitivity between the two polymer systems. I will say, though, that that last bullet point is mostly associated with troubleshooting. You can probably clean it up and get a good signal-to-noise ratio. Following some of the steps we outlined in the first webinar in our series. So please go back, revisit that webinar, and see if some of those points will help you obtain a better signal. So at the moment, we're going to explore these first 4 bullet points in a little more detail. Okay, you've got an existing system, but you want to amp up the sensitivity. What should I be looking at, or what should you be doing? Number 1, I've got here protocol optimization. Have a quick look at the antigen unmasking technique you've got. If you're using a microwave, maybe you're starting to get some inconsistency in the staining. Maybe that needs to be recalibrated, or you want to go to a different system, such as a pressure cooker. Maybe you could use a different buffer or a different salt, maybe a different pH. Instead of citrate, move to EDTA. Maybe that could give you a greater unmasking or retrieval and give you a better staining intensity. Another thing to look at, make sure you've got that primary antibody titer running. So this is where you go back and revisit those positive control sections. But if you've got an existing methodology, but you just, and those points are worked out and you're happy with them, they're established in the SOP, one of the easiest things you can do is swap out that end detection reagent, that enzyme substrate. And what I've got here is some examples, or an example, I should say, of 2 of our products. Vector DAB, here's the catalog number, and Vector ImmPACT DAB. Now we've got serial sections here. All things were the same in the workflow up until the point of when we added DAB. And you can certainly see that the ImmPACT DAB gave a much more heavier deposition of color, much more robust staining, which certainly aids in looking at weaker expressed antigen and a greater contrast with the underlying tissue architecture and morphology. So certainly this could be a very simple way to increase sensitivity. You can keep the existing workflow, the tissue prep, primary antibodies, secondary reagents, and just simply swap out this last color development. And this could certainly help increase sensitivity instead of your 1+ to maybe 3+ or something like that. If you've done all of these things, certainly then you may want to introduce an additional step into your methodology. And the idea here would be to increase the amount of enzyme that is present and therefore get a greater reaction with the enzyme substrate. You will remember from the cartoon I put up initially, the workflow, that one-step methodology. This one here incorporates an amplifier antibody, an unconjugated secondary that is then leveraged by a tertiary enzyme. You get more enzyme introduced, a greater reaction. This particular slide here shows a non-avid and non-biotin based system, and there are other multi-step methods. And this is one I've got here, is a avidin or streptavidin biotin based detection methodology. Now, this is an extremely flexible and modular system whereby if you did have different species of primary antibody, you can easily swap out a biotinylated secondary antibody and retain the enzyme avidin or streptavidin conjugate and the end part of the workflow here. So again, very modular, very flexible methodology. And it could certainly factor into reducing optimization and costs at your end if you want to increase the sensitivity. If you want to try and hit it out of the ballpark with some sensitivity, some investigators have been using a tyramide signal amplification system in combination with an ABC kit. Just bear in mind, though, that this will take a substantial amount of time to work up, to optimize. There will be associated costs with that, and a higher degree of potential, I would say, of developing background, and hence a poor signal-to-noise ratio. You will have to also run additional controls in there to make sure you're not picking up some false positives. But nonetheless, having said that, this just gives you some examples of how to increase sensitivity. Okay, let's pivot a little bit and just say you do have an existing methodology, but you know what? The PI says, hey, let's change tissue types, and we'll see if the antigen is expressed. Instead of, say, lung tissue, let's see if it's expressed in these kidney sections that we've got here. And certainly you might be scratching your head going, oh, gee, what should I be doing here? You know what? Take a deep breath. It's all okay. Keep the detection reagents the same. All you really want to do is just run a primary antibody titer series on this new tissue, because the antigen may be expressed at different levels in this new tissue, but keep the detection reagents the same. I will say that if you do swap tissue types, you may need to factor in some additional blocking steps based on the methodology you're using. So just keep that in mind. It is a little different, though, different story, when you do change species. For example, I've got here, amphibian species. You're getting some great results there, but you're expanding the study and you want to look at it in, say, a rodent species or other mammal species. This is where you're going to have to take a serious look at the current methodology. Number one, check that the primary antibody will recognize the target antigen in that new species of tissue. And secondly, just check that the secondary detection reagents do not present a cross-reactivity issue with that new tissue. For example, if you're using, say, a mouse primary antibody and an anti-mouse secondary detection reagent on the frog tissue, it may cause a big problem if you're going to use that, apply that to mouse and or rat tissue, and you're going to have to look at alternatives. But for the rest of the detection methodology, try and limit the variables to cut costs and optimization requirements. Okay, so you still have this existing methodology, but you want to explore maybe a different clone of the same species. Instead of one mouse primary, you want to look at a different mouse primary antibody, or indeed maybe using a mouse primary against a different target antigen. This is a little bit of plug and play. You'd substitute in a different primary antibody, but keeping the detection reagents the same, but just be aware, are there any caveats you might need to be aware of in order to maximize signal intensity with that given primary antibody? Different antigen retrieval, pHs or salts or things like that. Run a titer or dilution series as well, and just make sure you have those positive control tissues in place. If you are looking at a primary antibody, this is point number 2, raised in a different species. So you're changing now from a mouse primary with an anti-mouse detection reagent to, say, a rabbit primary antibody or a goat and introducing a secondary detection reagent with that. You know, number one, again, importantly, ensure the primary antibody recognizes the target antigen in that specimen. A lot of investigators, I know I've said this a couple of times, but a lot of investigators waste an inordinate amount of time without actually asking that question, and they certainly spend time and money on not addressing that, and that would go a long way to ensuring success for them at the get-go. And certainly then determine the appropriate secondary detection reagents. But if you are looking at multiple different antibodies that I mentioned earlier on, look for these flexible modular systems like the ABC reagents I mentioned earlier on, whereby you can easily drop in a different biotinylated secondary antibody. Look for universal reagents like anti-mouse and anti-rabbit, or indeed anti-mouse, rabbit, and goat secondary detection reagents, just being aware of potential species cross-reactivity. And there's also highly absorbed reagents available as well. All right, let's pivot a little bit now to multiple antigen labeling. This is a pretty big topic and almost a webinar in itself, which I'd probably say it is. So I can't really do it justice here, other than if you are looking to introduce a second or a third antibody on the same tissue section, the main considerations would be the choice of detection enzyme and the enzyme substrate combinations that you're hoping to use, and potential cross-reactivity between the secondary labeling reagents. But what I've got here, for those looking to maybe do a double labeling made easy, I've got one scenario here whereby, let's just say in your existing system, you've got a mouse primary antibody, you apply a goat anti-mouse peroxidase. This is your existing system, and you're developing with DAB. You're pretty happy with this, and you're thinking, you know what, I wanted to look at a different antigen. And you introduce a rabbit primary antibody. You apply an anti-rabbit alkaline phosphatase, different enzyme conjugate, and then apply a contrasting colored enzyme substrate at the end to give you a nice contrast. These you can mix together because there's not going to be any cross-reactivity. Importantly, these secondary detection reagents are raised in the same species, allows you to use the same serum. Let's have a quick look at another scenario. You've got this same mouse primary, you're using the same goat anti-mouse peroxidase and developing with DAB, but in this case for a double label, you apply a goat primary, apply a rabbit anti-goat alkaline phosphatase, and the same Vector Red at the end. Just note that there will be cross-reactivity between these two reagents if you mix them together. So you wouldn't be able to mix them together like the cartoon I've shown here. You'll have to run these sequentially. It will take you more time, but give you a much cleaner result. So these are considerations when looking at introducing double labeling. Okay, quick word about enzyme substrates. Why would I want to select one over the other? Well, as we showed earlier on with the DAB, sensitivity, and certainly moving from possibly a powder or a tablet format to a liquid format would potentially generate a greater degree of sensitivity and be an easy increase for you in terms of sensitivity from your 1+ to your 3+ or something like that. Have a look at the precipitate characteristics. The peroxidase giving more of a discrete, heavier deposition, whereas the alkaline phosphatase more of a diffuse, more of a translucent to a transparent look to them, which may be advantageous if some of the underlying morphology and architecture is very important. Maybe you're trying to avoid high levels of endogenous enzyme activity. You're struggling with blocking a lot of peroxidase for whatever reason, a highly vascularized prep. You know, you can simply sidestep that and go with an alkaline phosphatase system. And certainly a choice of color. Those of you who are familiar with DAB, you know, you'd have a hard time trying to differentiate DAB from the melanin deposits, I should say, showcased in this image. This is where a different colored substrate would be recommended. Solvent resistance, this would be whether or not you'd use something like AEC, an aqueous base media, compared to a more permanent mounting application, just be aware that there are time and cost concerns in going with permanent mounting application. Importantly, look at the scope of the project. Do you need to archive or store the material? If so, go with the permanent mounting application. If you're simply going to apply a cover slip and then image it and toss the slides, just go with an aqueous-based media. There's no reason to run the extra time and effort required for permanent mounting, even though this permanent mounting does give a better resolution of the target antigen. We did touch a little bit on single or multiple antigen labeling, so I'm going to leave that alone. But for those of you who might be wanting to dip your toe into both light field and fluorescence microscopy, there are some of these enzyme substrates that do happen to have the property of fluorescing as well as being highly visible, of course, under the light microscope. And you can do double fluorescent labeling with some of these substrates. Okay, let's finish up very quickly here with some cost considerations. Have a look at the, I will say, the assay volume and frequency. Are you going to do like 5 slides once a week? If that's the case, then go with the ready-to-use reagents. That's a lot more convenient. You don't need to be an expert in immunohistochemistry to get that up and running. But if you're maybe a grad student at the first couple of weeks into a 4-year project where you're going to be doing a lot of slides almost every day, you're going to want to develop a little bit of skill and knowledge about it. It will take you a little bit more time, but certainly there are some cost considerations or advantageous cost considerations associated with that. I can't answer this question for you, but just to dovetail back to our original webinar in this series, the goal of immunohistochemistry is to get a clear, unambiguous view of the target antigen, consistent, reproducible staining. All right, at this point, I'd like to thank you very much for your time. I hope you have enjoyed the webinar series and you found it educational and informative. Please stay with us and stay in tune for our next webinar series commencing early next year. And at this time, once again, thank you very much, and I will open the floor up to any questions you may have.
All right, thank you, Craig, for that great presentation. We'll start the Q&A session. The first question, for double labeling, my host species of primary antibody is the same. What should I do?
Yeah, for immunohistochemistry, we've found actually that if you run those sequentially, and what I mean by that is you'd apply the first primary antibody. Let's use the example of a mouse primary antibody first. You'd apply an anti-mouse, let's just call it a peroxidase-based detection methodology, and then develop with the enzyme substrate. That will be your first label. So that's from primary right through to color development. Then you would apply the second primary antibody, again a mouse, then the anti-mouse, it could be alkaline phosphatase or peroxidase, and then develop that second enzyme substrate. So performing that sequentially works very well with immunohistochemistry. You know, the enzyme substrates precipitate out or oxidize as solids on the tissue section, and what they do is almost form a bit of a protective layer around those detection reagents and prevent that subsequent interaction from the second detection reagents binding or cross-reacting with the first labeling reagents. So that's the methodology for immunohistochemistry. It would certainly be very different for immunofluorescence. So just take note of that, but that's generally what we'd recommend. Indeed, in the presentation earlier on, I did show an example of 2 HRP substrates. I believe it was DAB and Vector VIP. They were actually performed using 2 mouse monoclonals, if I remember correctly. So it just gives you a bit of an idea, and they were in very close position to each other. So yeah, certainly sequential labeling would be recommended.
Okay, thank you, Craig. The next question is about secondary antibodies. Can you elaborate more on the interaction of the secondary antibodies, either as the target species or host species? Does this only apply to IHC or other techniques?
Okay, so, well, I'll interpret that as being a secondary antibody interaction with the primary or the tissue type. So, for example, let's just use the example I used a little bit earlier on, say a mouse primary antibody being applied to a mouse specimen. I hope I'm interpreting this question appropriately, whereby you're using an anti-mouse secondary antibody to recognize that unconjugated mouse primary antibody. That secondary antibody, of course, would bind to not just the mouse immunoglobulin, whether it be a mouse IgG or IgM, but also inherent IgG or IgM in that tissue preparation. And that's certainly the species-on-species cross-reactivity. So that would be problematic. And there are commercial blocks available for that if you want to go that route. If you want to avoid that, you could look at changing out the species of the primary antibody. So you could then use a different secondary antibody, one that would not cross-react with the tissue IgG, or indeed you could consider conjugating that primary antibody either directly with an enzyme or a small hapten molecule like biotin, and then follow through with an avidin or a streptavidin conjugate and avoid having to use the, in the case I used, an anti-mouse on mouse tissue or a secondary antibody. So hopefully that clears that up. In terms of the host species, I did mention a little earlier on about, goat anti-mouse, HRP, for example. You could certainly use a range of different species of secondary antibody. If the idea is to bind to a mouse primary, you could use a goat anti-mouse, you could use a horse anti-mouse for a single label. It doesn't matter what that species of the secondary antibody is raised in. And indeed, the idea would be to use serum from the same species that the secondary antibody is raised in as a general blocking reagent. So it doesn't really matter. Just be aware, though, of any potential cross-reactivity with, a second or a third primary antibody or detection reagent that you might introduce. Hopefully that answers the question.
Great, thank you, Craig. The next question, how should I select a VECTASTAIN ABC kit versus a polymer system?
Yeah, these are parallel detection methodologies. The ABC system certainly has a track record of being widely used in many applications, whereby it exploits the interaction of, or high affinity of, avidin and biotin. And as I mentioned earlier on, it's a very modular, flexible methodology, available in a lot of different formats, both streptavidin and avidin based. So indeed, it'll allow you to swap between methods. If you're looking at a biotinylated reagent in Western blot and ELISA, you could certainly use those same reagents for immunohistochemistry. But nonetheless, what I would say is that there are some tissues, such as kidney and liver, that do sequester detectable levels of biotin, and one of those systems may be, one of those tissues may be problematic when using an avidin biotin based reagent, and that would require an extra blocking methodology. Indeed, what you could do then would be to choose a non-avidin biotin based system, such as a polymer based system, the ImmPRESS reagents I showed earlier on, as a complete or comprehensive kit format, that don't have a biotin or avidin component in them, and therefore there wouldn't be a concern for those reagents detecting that biotin in those tissues, and certainly equivalence in terms of sensitivity in a lot of cases. So it depends a little bit on the tissue type you're looking for and maybe the scope of the project. All right.
All right, thank you, Craig. The last question is, can I use a universal secondary antibody, for example, anti-mouse and anti-rabbit, on rat tissue?
The quick answer is yes, but you want to be a little aware of the fact that the anti-mouse component of that universal antibody would have some % of cross-reactivity with rat IgG. Now, if the tissue itself doesn't exhibit any detectable IgG, then there wouldn't be a concern in applying that. Maybe the tissue is, a cell monolayer, or or something, maybe a perfused fixed specimen that's washed out, almost all of the IgG, then you can apply that mouse rabbit secondary antibody. But just be aware that if you've got a vascularized prep, it's been immerse fixed, and there is detectable levels of rat IgG in there, that anti-mouse component of that universal secondary antibody will cause background staining in that rat tissue. So you will have to look for something like an anti-mouse rat-adsorbed secondary, or indeed, if you are using a rabbit primary, look for a defined anti-rabbit secondary antibody.
All right, thank you, Craig. Our time is up for this webinar. Thank you for joining us this time. Please don't miss our next webinar series in early 2021, which will include bioconjugation and lectins. We will send you the entire recording of the webinar through email. And if we did not answer your questions in this webinar, we will answer them personally later. Please feel free to follow up with our email, technical@vectorlabs.com. Thank you for joining. Have a great day.
Thank you very much. Bye.
All right. Bye-bye.